That gonna work? Yeah. All right. So, hello everybody. Quick note here. If you haven't noticed, I updated the schedule on Blackboard. Okay. So it reflects kind of where we are versus where the original schedule suggested we would be. And you may guess we are not on schedule. So everything's gonna be kind of revamped. Originally, the second exam was going to be Thursday before spring break, so in 1 week. We're definitely not getting there. So the exam is going to end up being the second week after spring break. Okay, so we are going to finish up the TCA cycle today. Next week, we'll talk about the mitochondrial electron transport chain and oxidative phosphorylation. Last bit of sugar metabolism, and that's glycogen, right? So glycogen is how we store sugars as energy. So we're going to talk about glycogen synthesis and glycogen degradation the Monday after spring break. And then you'll have a week to prepare for the exams. So I don't want the exam to be the next class period. So the next class period will jump into a new topic that won't be on the exam, but that way we kind of have more time to do the problem set and to ask questions and think about stuff. So I think the date's the 28th. I think so, but verify that. But it's the second Tuesday after spring break. Okay. Okay. So today we're going to finish up the TCA cycle, and we got through the chemistry of the pyruvate dehydrogenase complex on Tuesday. We're going to review all that today, and then we're going to finish up what we want to get through with the TCA cycle. So as a reminder here, the point of the TCA cycle is to finish oxidizing the carbons that start at glucose, right? So 6 carbons of glucose ultimately split up into 2 equivalents of pyruvate. Those 2 equivalents of pyruvate then will proceed through the pyruvate dehydrogenase complex. Okay, this is an example of oxidative phosphorylation oxidative decarboxylation. We decarboxylate the CO2 group on pyruvate, and we also oxidize the carbons. So when we lose CO2, that carbon gets oxidized and those electrons are given to NAD+. Okay, so oxidative decarboxylation, and we end up with acetyl-CoA. Acetyl-CoA, a 2-carbon unit, then enters into the TCA cycle. And the point of the TCA cycle is then to finish oxidizing those 2 carbons. So 2 carbons come in, 2 carbons go out in the form of carbon dioxide. Okay, and I mentioned that a really important theme in the TCA cycle is focusing on the alpha keto chemistry. Okay, so remember, alpha keto simply means the alpha carbon, the one that's right next to the highest priority carbon, our carboxylic acid, that's our alpha carbon, and it's a ketone. So the chemistry of the TCA cycle focuses around this alpha keto group. Once we make this alpha keto group, there are 2 fates. One is oxidative decarboxylation. Pyruvate gets oxidatively decarboxylated. And another alpha keto compound, alpha ketoglutarate, gets oxidatively decarboxylated. This produces CO2, generates 2 electrons that are stored as NADH. Okay? The other fate of an alpha keto compound is the starting point for the TCA cycle. We're going to see that this alpha carbon, this alpha keto, is the nucleation point for the addition of 2 carbons onto oxaloacetate to give us citrate. That's how acetyl-CoA enters into the TCA cycle. Okay, so the fate of the alpha keto compound is either oxidative decarboxylation or entry point into the TCA cycle. We make the alpha keto compound either by dehydration, which we saw in glycolysis. We have 2-phosphoglycerate, which we dehydrate to give us phosphoenolpyruvate. That is an alpha keto compound, right? Or we can oxidize an alpha alcohol to give us an alpha ketone. And this is the chemistry that we really see happening primarily in the TCA cycles. We oxidize an alpha alcohol. Okay, so let's see how this works in. So here's our TCA cycle, including pyruvate, and I've shown the 3 alpha keto compounds: pyruvate, oxaloacetate, and alpha-ketoglutarate. And note here that I have both alpha-ketoglutarate and pyruvate highlighted in red and the oxaloacetate in blue. And that's because the fate of pyruvate and alpha-ketoglutarate is identical, oxidative decarboxylation. And the chemistry in this step is literally identical, okay? The enzyme that catalyzes the oxidative decarboxylation of pyruvate is pyruvate dehydrogenase. The enzyme that does it for alpha-ketoglutarate is alpha-ketoglutarate dehydrogenase. The chemistry is identical, absolutely identical. The only difference is what hangs off of the alpha carbon. For pyruvate, it's a methyl group. For alpha-ketoglutarate, it's 3 other carbons. That's the only difference, okay? So we are going to focus in on the chemistry of the pyruvate dehydrogenase complex, and you can apply that to alpha-ketoglutarate dehydrogenase because it's the same chemistry. Put an R after the alpha carbon, and everything's the same, okay? Alpha-keto, or the oxaloacetate, is our other alpha ketone. That's the point of entry for acetyl-CoA, right? So this alpha ketone ends up getting attacked by the beta carbon of acetyl-CoA so that we can make citrate. Okay? We said that to make the alpha ketones, to make our alpha keto compounds in the TCA cycle, this comes by oxidizing an alpha alcohol. And in both cases here, that's what we see. We oxidize malate, an alpha alcohol, to give us oxaloacetate. We oxidize isocitrate, an alpha alcohol, to give us alpha ketoglutarate. Okay, so 4 steps in the TCA cycle we understand purely based on the production or the degradation of our alpha ketones. Focusing on the alpha ketones, everything kind of falls into place. So the 2 steps preceding them are oxidation steps to produce the alpha ketones. Okay? To make our alpha alcohols, we either hydrate a double bond to make an alpha alcohol, which we're seeing here. So fumarate becoming malate is hydrating a double bond to give us an alpha alcohol, or we are rearranging a beta alcohol. Citrate, citric acid, is a beta alcohol, right? So this is the beta carbon, that center carbon. That's a beta alcohol. We isomerize it to give an alpha alcohol, which can then be oxidized to give the alpha ketone. Okay? To make fumarate, an alkene, we oxidize an alkane. So we have succinate, we oxidize that to give us the double bond, which is needed to hydrate the double bond to give us the alpha alcohol, which can then be oxidized to give us our alpha ketone. Right? So all of these steps are leading up to being able to make our alpha ketone. The remaining steps are a transfer reaction. So our alpha ketoglutarate, we oxidatively decarboxylate, and we end up with succinyl-CoA, right? The energy of hydration, of hydrolysis of that thioester bond is enough energy to produce an equivalent of ATP. And that's what we see in this very final step, right? So going from alpha ketoglutarate to succinate could happen in one step. It very easily could, just like we saw in glycolysis, right? We could bypass this thioester. But by forming this thioester, we end up making a high energy compound that can be utilized to make an ATP for us. Okay? So if you focus on the alpha ketone and what's needed to make the alpha ketones and regenerate the alpha ketones, I think the TCA cycle really makes sense. I mentioned yesterday that we're going to focus primarily on 4 steps, okay? And we started in on the pyruvate dehydrogenase complex already, okay? The other steps, isomerization of citrate, oxidation of succinate, hydration of fumarate. These things are all things that are much more straightforward and chemistry that you've already really kind of seen when we talked about glycolysis. Okay, so there's nothing especially surprising. So the pyruvate dehydrogenase complex we mentioned Tuesday is this really massive complex that you can very easily see on using cryo-EM. Okay, each of these little blobs, each of those little dots within the bigger blob is an individual enzyme. Okay, there are 3 different active sites which catalyze 3 distinct reactions. So if we think about the overall chemistry that has to happen to oxidatively decarboxylate pyruvate, right? We start with pyruvate. Decarboxylation of pyruvate produces carbon dioxide. So this carboxylic acid carbon gets oxidized, and as a result, this what was the alpha carbon, this carbon gets reduced. Okay, we generate a carbanion. It's holding on to 2 electrons. That then gets oxidized. Those 2 electrons are given to something else, creating a carbocation, which can then be transferred onto acetyl-CoA. Okay, so we look at these intermediates, our carbanion, our carbocation, and we immediately know that these can't be stable. So this must be facilitated by something else. Whenever we create these unstable intermediates, it's very likely that they are covalently anchored to something else. Okay, and for the pyruvate dehydrogenase complex, those something else are cofactors that some we've talked about, one we haven't. So one of the cofactors that holds on to this 2-carbon unit is TPP. We've seen it before. Remember, this is thiamine pyrophosphate, and this forms a stable carbanion on the carbon on the top there, right? So this can easily be deprotonated, forming a stable carbanion. That generates a really nice nucleophile for some of these for nucleophilic attack. The other notable thing about TPP is that we have this positively charged nitrogen, which we've seen before, is a really great electron sink. Okay, this is going to be important. We're going to see that the carbanion gets linked up to TPP. Lipoic acid's the other one. Right here, we're showing the oxidized form, the disulfide form of lipoic acid. It can also get reduced to have a dithiol. So this double bond, or that sulfur-sulfur bond is not there. Instead, you have an SH and an SH. This is the other cofactor that can make a covalent bond with our acetate group, with our 2-carbon group. Okay, it's going to hold on to the oxidized form of carbon. Okay, ultimately, all of this happens in 3 different active sites: E1, E2, and E3. And the names kind of describe what happened. In the first step, this is where we decarboxylate and oxidize our carbons. Okay. In the second active site, the dihydrolipoyl transacetylase. The idea here is, in this active site, we are taking an acetyl group, putting it onto our lipoic acid, and then removing it from our lipoic acid. Okay? And then in the third active site, this is the dihydrolipoyl dehydrogenase. So at this point, the only thing left to do is to transfer electrons from the reduced lipoic acid ultimately to the NADH cofactor. Okay, so we talked briefly about how this works and how this complex is arranged. Each of these spheres represents an individual enzyme. Okay, the yellow is E1, that's the pyruvate dehydrogenase part of it. In the middle, the red is E2, that's the dihydrolipoyl transacetylase. That's where our lipoic acid is covalently anchored. And the cool thing about this is it's kind of like a Tarzan unit, right? It's a vine that lets the C2 group swing between different active sites. And then the green is E3. This is our dehydrogenase component. This is where we are transferring electrons from the reduced lipoic acid through flavin onto NADH. Okay, so let's talk through this chemistry, and then I'm going to go ahead and work through the mechanism, and y'all can copy it if you didn't finish up on Tuesday. Okay, so what I want to do here is focus on electron flow. All right, so here's our 3 active sites. Note here that the E2 is shown as a dimer. One of them is where the lipoic acid is anchored to, and the other one is the actual active site that we use. Okay, so there's 2 of them that are functional here. So the first thing that happens is that pyruvate enters into the E1 active site. We get oxidative decarboxylation, so we lose carbon dioxide. The electron, the carbon that leaves as CO2 gets oxidized. Okay, and we end up with this carbon right here, this central carbon. That carbon started as a ketone, it's now an alcohol, right? So that carbon has been reduced. It's that carbon that's holding on to 2 electrons at this point. The carbon and carbon dioxide gets oxidized. That central carbon now is the one that's reduced. It's holding on to electrons. In the next step, we have our at this point, we also have a covalent bond between TPP and our 2 carbon unit. This triggers our lipoic acid to move into the E1 active site, which triggers transfer of the acetyl group from TPP onto lipoic acid. We see that when this happens, we end up starting off with a double bond or a single bond between our sulfurs, and that single bond gets broken. So our sulfurs get reduced. The electrons are transferred from that carbon onto the sulfurs. The carbon gets oxidized, the sulfurs get reduced. Okay? In the next step, we take our C2 unit, our 2 carbon unit, and we transfer it from our lipoic acid onto coenzyme A. So coenzyme A, a reactive thiol comes in, simply replaces that sulfur of lipoic acid. We break the thioester bond with lipoic acid, and we make a new thioester bond with with coenzyme A. Acetyl-CoA exits the active site. One of our products is now made. What we have now is our lipoic acid that needs to be regenerated, right? We have lipoic acid holding on to 2 extra electrons, but we need to reoxidize it so that it can function as it needs to up here. Okay? So these electrons are first passed through FAD. So FAD in the oxidized form gets reduced to make FAD2, or FADH2, and our lipoic acid starts reduced, gets oxidized. Okay? So the electrons ultimately that left the carbon from CO2 are now on this FADH2 cofactor that's covalent, that's not covalently, but very strongly anchored into this this E3 active site. And the last step is they get transferred onto NADH. Okay, so electrons are passed through flavin to be given to NADH. So tracking this all the way, we start with pyruvate. This carbon gets oxidized and we produce CO2. Those electrons are originally held onto by the carbon of our acetyl group. They're then passed to the sulfurs of lipoic acid, then passed to flavin, and then passed to NADH. Okay, so that's the flow of electrons here. The CO2 carbon's oxidized, ultimately NAD plus gets reduced to make NADH. Questions about that? Okay, so now on Tuesday I gave you like the 15 minutes at the end of class to work through this mechanism, and most of you all got through at least the first couple steps. So let's go ahead and work through this together just to reinforce. Okay, so step 1, we have pyruvate entering into the active site. Okay, things to notice is that we start off with our alpha ketone, we end up with an alcohol here, and we have a covalent bond between our TPP cofactor and that central carbon, which was the alpha carbon, but yeah, so it still is the alpha carbon at this point. Okay, so TPP comes in, it's the nucleophile, that alpha carbon is the electrophile, the electrons flow up to the oxygen, and we pick up a proton here. We end up with this alpha alcohol with the alpha carbon covalently linked to our TPP cofactor. This, I hope, is fairly straightforward. It's a nucleophile attacking an electrophilic alpha carbon to produce an alpha alcohol instead of an alpha ketone. In the next step, we have this alpha alcohol and we decarboxylate it. Remember, decarboxylation always happens in the same way. We have this carboxylic acid group. This bond is going to break and the electrons are going to flow somewhere. And TPP presents a really nice electron sink. So the electrons are going to flow all the way over to our electron sink. So those electrons move over to make CO2. This bond gets broken. We move the electrons over here to make a double bond, and then push them all the way over to our electron sink nitrogen. In the next step, remember, with these electron sinks, it's very common that as soon as we put electrons on the electron sink, we now want to reverse it and move them somewhere else. Okay, and if we look carefully here, what we notice is that we are putting them onto that central carbon. We're going to temporarily make a carbanion, which can pick up a proton to be stabilized. So we are taking the electrons on this nitrogen, we are reforming our double bond, and then we're moving the electrons of that double bond, that pi bond, over to that central carbon to make a carbanion. And then you could easily show this picking up a proton if you want to. But at the end of the day, that proton can come from water, it can come from a general acid on the active site, whatever it may be. Okay? We've picked up that proton, we've made this intermediate, we're completely decarboxylated. We have our C2 unit, our acetyl unit, happily anchored onto TPP. We know that in the next step, in the next active site, we want to transfer this onto our lipoic acid cofactor. So lipoic acid comes in. It's in the oxidized form. Okay, note here that it's the carbanion form of this carbon that's reactive. Okay, so we have our carbanion. What we note is that we need to make a bond between this carbon that I point out and one of these sulfurs, and you could choose either one. The way that I drew it is kind of backwards, but we'll stick with it. So the carbanion attacks here. That pushes the bonds between the sulfurs up to the other sulfur. This sulfur picks up the proton there on that alcohol. The electrons in the OH bond move over to make a double bond, and then we break that bond there. So it's really just kind of a rearrangement of electrons here. There's lots happening here all at one point, like all at one time. The reaction is initiated by that carbanion. It attacks one of the sulfurs. That breaks the sulfur bond, that breaks the disulfide linkage. That other sulfur has to pick up a proton. Okay? It's easiest to imagine it coming from the alcohol on the original carbanion, because that proton has to go away to reform our carbonyl. We make our carbonyl, and that breaks the linkage between the acetyl group and TPP. We have effectively transferred the acetyl group from TPP all the way over to lipoic acid. And the other thing that's happened here is electron density has moved off of that carbon. We've reoxidized that carbon, and the electrons are now being held onto by the sulfurs in our lipoic acid. In the next step, coenzyme A comes in, and this is fairly straightforward. So we can imagine a general base deprotonating that sulfur on coenzyme A to make it a little more reactive. We attack the carbonyl. You can imagine an intermediate being formed if you want to, it collapsing, and we move the acetyl group from lipoic acid over to coenzyme A. We have now formed acetyl-CoA, one of the products of this complex, one of the products of this reaction. The next two steps simply involve transferring electrons from reduced lipoic acid through flavin all the way over to NADH. Okay? And the chemistry here is something you've absolutely seen before. Okay? We know that whenever we're dealing with FAD, if we want to reduce it, we need a hydride source. The hydride is going to come from lipoic acid. So let's imagine a general base here that reaches out, deprotonates one of the sulfurs. The electrons in the SH bond move over to make a bond with the sulfur, and then we have hydride exiting. Okay, so we deprotonate one of the sulfurs. The electrons in the SH bond make an SS bond, and then the SH bond on the other sulfur has to be broken. So we have hydride exiting, which can then attack and move through flavin like we know it has to. And we end up with reduced FADH2. Okay, again, this hydride movement, these electrons moving through FAD, should not be surprising. It's something we've seen before. Same with this next step. In the next step, we are taking hydride off of flavin and giving it to NAD plus to make NADH. So we just want to reverse this. So propose deprotonating either of the nitrogens on flavin. I don't really care which you use. I'll go ahead and deprotonate the top one. We deprotonate that. The electrons move back through flavin, ultimately generating hydride. The hydride attacks at the position of NAD plus that we know it has to, giving us reduced NAD plus. We have NADH. Questions about that? Okay, remember, I mentioned that this chemistry is identical to the succinate dehydrogenase or the alpha ketoglutarate dehydrogenase mechanism. Everything's the same. The only thing that's different is up here, instead of having just a single methyl group, we have other things. So you can imagine this just being an R group hanging off. That R is the rest of alpha ketoglutarate. The chemistry otherwise identical. If you know the PDH process, you know the alpha ketoglutarate dehydrogenase complex process. Okay, so let's talk about citrate synthase. This is the entry point into the TCA cycle. Okay, the idea here is we are activating the beta carbon of acetyl-CoA to be a nucleophile, attacking oxaloacetate to make this cityl-CoA intermediate. Okay, so without really going into the chemistry here, the general idea is this carbon gets activated as a nucleophile. It attacks at the alpha ketone of oxaloacetate. We pick up a proton. We make this intermediate where we have an alpha alcohol. In this case, it's actually a beta alcohol. But we make the alcohol equivalent of it, and we formed a new bond between acetyl-CoA and oxaloacetate. So we've gone from a 4-carbon oxaloacetate to a 6-carbon citryl-CoA. And then the next step, we hydrolyze the thioester bond, a very exergonic process to generate citrate. Okay? The one thing I want you to think about here is hydrolysis of acetyl-CoA. Hydrolysis of that thioester bond is very exergonic. This enzyme can easily do it, right? This enzyme stabilizes the transition state for this hydrolysis reaction. It does, right? It does it in the second part of this reaction. So the question I have for you is, how does the enzyme prevent this hydrolysis from happening at the beginning of the reaction? This is the same type of question, the same type of thought process you should be thinking about how kinases prevent hydrolysis of ATP. How do kinases prevent hydrolysis of ATP so that they can facilitate the phosphate transfer instead of hydrolysis? So that's how it stabilizes the transition state. Like that's very possible way of stabilizing the transition state. But that doesn't prevent hydrolysis. The idea is that hydrolysis of these high energy bonds is spontaneous. And if the enzyme is capable of decreasing the activation energy to make it kinetically viable, then there has to be a way of preventing this second step from happening first, because this is the spontaneous step. Obany. Yeah, this is where we have our induced fit mechanism, right? So the idea here is that the oxaloacetate must bind first to get into place before acetyl-CoA binds. And once if the oxaloacetate is already in place, then water is excluded, so we can't hydrolyze acetyl-CoA until the reaction is at the right point. Okay, and that's exactly what happens. Oxaloacetate binds first. That causes a structural change, which shifts this, like you can like see the motion here. This swivels over, that closes the gate. Acetyl-CoA can then bind. When we make our citryl-CoA, that induces even another structural change, which then allows water back in. Okay, So the water can't get close to the active site until we make our citryl-CoA intermediate that we see here. Okay? Whenever we're talking about preventing certain chemistry, right? Especially preventing hydrolysis of otherwise very spontaneous reactions, the answer is almost always going to be induced fit, right? The only way to do that is to have some sort of a structural change that can exclude water to prevent the undesired chemistry. Okay, so the way this works is reasonably straightforward. Okay, so we have our complex already formed where we have oxaloacetate and we have acetyl-CoA bound in the active site. Okay, we have three important amino acids present here. The histidine we see making a hydrogen bond with the alpha ketone, right? So oxygen making a hydrogen bond with the histidine. Another histidine we see is making an important hydrogen bond with the carbonyl of acetyl-CoA. And then the aspartic acid is in close contact with the beta carbon of acetyl-CoA. Okay, and it's this aspartic acid that initiates the reaction. It deprotonates that carbon. Okay, so this reaches out, picks up a proton. This proton moves over to aspartic acid. The electrons in the CH bond move over to make a double bond between carbon 1 and carbon 2. And this is stabilized by the pi bond in the carbonyl reaching out and picking up that proton from histidine. So we end up making this enol intermediate where we have this alkene and an alcohol. So we make this enol intermediate. Okay? By doing this, we prevent the need to make a carbanion on that beta carbon, right? Because in the previous slide, let me flip back here real quick. In this previous slide, I kind of mentioned that you imagine this being a carbanion, because ultimately this ends up being the nucleophile. But by creating this double bond, we prevent the need to make a carbanion, because now when this enolate, this enol reverts back, the electrons in the double bond don't pick up a proton again, but instead they reach out, and they're the electrons that are the source of the nucleophile to attack the alpha ketone on oxaloacetate. So we have deprotonation, we reform our carbonyl, and then these electrons in the double bond reach out to make the bond with oxaloacetate. And then this picks up a proton so that we ultimately create an alcohol and we don't have the ketone anymore. Okay, this gives us our citryl-CoA intermediate. So you see we have this new bond between the beta carbon of acetyl-CoA and the alpha carbon of oxaloacetate. So we have that new bond. We now have a 6-carbon intermediate. In the next step, we simply need to hydrolyze the coenzyme A off of this to end up with citric acid. Okay, and again, the important thing to recognize here is it's this part, it's the production of this intermediate that induces a big structural change that lets water come in, and once water's in, then we can hydrolyze that off, and that chemistry is not surprising. Water's a nucleophile, it attacks at that carbonyl, and we break the CS bond. Okay, questions? All right, moving on. Aconitase. So this is a really, really cool enzyme. And the reason I think it's cool is because it's actually a metal, it's a metal sensing protein. It moonlights in eukaryotes as an iron sensor. There's a couple different isoforms. One is present in the cytoplasm, the other in the matrix, right? The matrix in the mitochondria, that's where this chemistry is happening. But the isoform that's present in the cytoplasm actually serves as a metal sensor, and that's really kind of cool, right? It serves as basically a transcriptional repressor. So that's just kind of a cool feature of this enzyme. But the chemistry we see here is isomerizing citrate to isocitrate. So we have this beta alcohol that we're converting to an alpha alcohol, right? So this central carbon is our beta carbon, no matter which direction you count from. That's 2 carbons away from the carboxylic acid, so this is the beta carbon. And we are moving this alcohol up to the the alpha position. So we're going from a beta alcohol to an alpha alcohol. And we do it in 2 steps. The first step is dehydrating citrate to make this double bonded intermediate, this cis double bond aconitate it's called, and then we rehydrate the double bond. Okay, and we're not going to really look at this chemistry beyond just saying this, but note that this dehydration is incredibly similar to dehydration of 2-phosphoglycerate to make PEP, right? For this to happen, we need a base to deprotonate that carbon, and ultimately the electrons need to move down here so that we can release water. But that's not favorable. Instead, we saw with the with enolase, right, dehydrating 2-phosphoglycerate, we saw that originally when this bond breaks, the electrons move up here, and we end up with this di-negative intermediate that's stabilized by a couple magnesiums that are present up here. The same type of chemistry happens here. We first go through this intermediate where the double bond is between C1 and C2, and then the double bond moves down here to where it needs to be to release the hydroxide. Okay? If you ignore this part, right, just kind of ignore all this and just call it R, the chemistry is exactly the same as enolase. And then when we add water back in, the same type of thing happens. Water attacks at the C2 position. Electrons move over here to remake our double bond, and then that's it, right? So it picks up a proton. So once you make that double bond, that cis enolate intermediate, a aconitate intermediate, the chemistry is straightforward. It's just then hydrating a double bond. All right. So isocitrate dehydrogenase. This one I think is part of your activity. So I'm going to let you work through it. But let's talk about the chemistry first. There's 2 steps. In the first step, we oxidize. It's not oxidation of NAD. So we oxidize this carbon here, and those electrons are given to NAD+. We generate NADH. In the second step, we then decarbonate. We decarboxylate. I can't talk, y'all. We decarboxylate this intermediate. So step 1 is the redox step. Step 2 is the decarboxylation. Okay. So I'll give you about 10 minutes here to try to work through this by yourself.